Hello everyone, welcome to today's presentation. My name is Axel Villagomez Rodriguez. I'm a student of Master Program in Life Science Innovation, and today I want to talk about, are natural products useful for cancer treatment? Specifically, the anti-proliferative effects of ferulic, coumaric, and caffeic acids in HepG2 cells by HTRT downregulation. First, to understand this research, we need to talk about the end replication problem. As you may know, for eukaryotic cells, when cell division occurs, the DNA at the very end of the chromosome cannot be fully copied in each round of the replication. This happens in these areas of the chromosome called telomeres, where every time the cell divides, usually what happens is that the DNA replication machinery cannot fully copy these regions. So there is a reduction of the length of these areas, which is associated with cell aging and replicative senescence. So over time, when we age, our telomeres become shorter and shorter, and this is a normal function of cells. However, we have a system to avoid this cell aging, which is mainly the main component is the telomerase, which is a ribonucleoprotein for the DNA repair system. This protein synthesizes the telomere particular structure, and this nucleotide repeats to protect the end caps of the eukaryotic chromosomes. However, it is important to note that this telomerase is commonly inactive in normal somatic cells. However, the telomerase activity and expression is considered a hallmark for the majority of human cancers because elevated levels of the expression and the activity of this protein has been observed in different researches. So, the question here for these authors was, if the inhibition of telomerase occurs, is it possible to increase sensitivity to anti-cancer drugs? So for that, they performed a little bit of research on the literature, and they found that there are different compounds that can have an effect on the expression and the activity of the telomerase. For example, here we can see the stilbene compounds. They found that they are able to control telomerase levels and or activity, which may be important in chemotherapy. For example, through telomerase activity inhibition, oxidative stress, or even apoptosis activation. However, there is limited knowledge of the effects of natural products on telomerase activity or expression in cancer cells. There are a few examples, like luteolin, which is a flavonoid, which is able to suppress the human telomerase reverse transcriptase, the HTRT gene, which is really important because it codifies the human telomerase. In this case, these researchers found this activity of luteolin in breast cancer. And there are also other molecules like these two, which are found in blackberry juice, that also showed in inhibition of the telomerase activity in HepG2 liver cancer cells. So even though there are some research about this topic, there's still a lack of knowledge of the effects of different natural products on the expression of this important protein. So in this case, the authors ask themselves if these phenolic acids that they selected can have like anti-cancer activity through inhibition of this gene expression, the human telomerase. They selected these three molecules because they have different number of hydroxyl groups in their ring. In the case of coumaric acid, we can see that they have three different variants, which is the ortho, meta, and para molecules, but they represent the same, with only one hydroxyl group. And the same case is for ferulic acid, which also only has one hydroxyl group, and caffeic acid that has two hydroxyl groups. They tested these molecules because they are found in a lot of natural products. These are quite common. And they wanted to know if they cause cytotoxicity, apoptosis, and downregulation of the human telomerase gene expression. For that, they use HepG2 cells. First, they tested the viability of these HepG2 cells with different concentrations of the phenolic acids, from 25 all the way to 800 micrograms per milliliter. And to test this, they used the MTT assay at different incubation times for 24 and 48 hours separately. For the apoptosis, they performed flow cytometry analysis using this detection kit with annexin and these markers. And for the gene expression analysis, they used two different concentrations of the phenolic acids, which are 600 and 1200 micrograms per milliliter. They isolated the total RNA, performed cDNA synthesis, and the detection, they used cybergreen and through real-time PCR using beta-actin gene as control. And the amplicon assessments were done using melting curve analysis. So, the first question was, for the experiments, if these phenolic acids cause reduced proliferation of the HepG2 cells. So, as explained previously, they use different doses of these three phenolic acids, and they measure the viability using the MTT assay. They reported the inhibitory concentration 50, which is the concentration when you observe reduced proliferation of cells by 50%. And as you can see here in the results, they reported that for the three phenolic acids, the concentration needed to reduce the proliferation of cells by 50% is decreased in a time-dependent manner. For example, if we take this caffeic acid, we can see that the dose required to reduce the proliferation by 50% is less when we have 48 hours and a little bit less when we have 72 hours. So these results suggest that the phenolic acids reduce the cell proliferation, but we don't know exactly with this assay if these cells are necrotic or apoptotic. We only know that the proliferation is being reduced. So for that, they presented this second question: if these phenolic acids cause reduced proliferation by apoptosis. So for that, they performed this flow cytometric analysis using the detection kit, and they reported these graphs where the interpretation is quite simple. If if all of the cell populations are represented here, it means that they are viable cells. If they are here, it means they are necrotic. In this area, it means that they are late apoptotic cells, and here they are at an early stage of apoptotic cells. So for their negative control, they observed that 96.8% of the cells were viable. For caffeic acid, they observed that mainly the cells were in this area, which is late apoptotic cells, and they represented a 98.4%. In the case of ferulic acid, they observed in this area also for late apoptotic cells 97.5%. And in the case of coumatic acid, they observed 92.9% in the same stage for late apoptotic cells. So coming back to the question, if these phenolic acids cause reduced proliferation by apoptosis, we have this summary of the results. As you can see, the cell number counted by percentage in negative control, again, is almost 97%. And for the treatments, they observe the most amount of apoptotic cells, and the necrotic ones is really low. So these results suggest that these phenolic acids cause the reduction in proliferation by an apoptosis mechanism. However, we are only observing here the result. We don't know exactly if they have an effect on the human telomerase expression. So according to their review, the many phenolic acids, not these ones, but many other phenolic acids have been detected to have these anti-cancer effects on different cell types. So according to the review, this also applies to these 3 phenolic acids, at least for the HepG2 cells, but we still don't know if this has an influence or is it involved in the expression of the human telomerase. So that takes us to the next question: if these phenolic acids affect the human telomerase expression. So for that, they performed the real-time PCR with the cDNA, the synthesis of the cDNA, and using beta-actin as control. So they performed 2 different doses, as you can see, 600 and 1200 micrograms per milliliter. We have the ferulic, coumaric, and caffeic acids. And as you can see, for some acids, if we increase the dosing, it seems that is dose-dependent, the effect. In the case of ferulic acid, the effect is a little bit reduced comparing to the lower dose. For coumaric acid, in this case, the effect is stronger. And for caffeic acid, it also loses a little bit of intensity on the inhibition of the human telomerase gene expression. So in this case, we can see that for 600 micrograms per milliliter, the caffeic acid is the one that has the strongest inhibition, followed by ferulic and coumaric acid. But if we increase the dosing to double, we can see that coumaric acid now is the one that has the strongest inhibition of the expression of the human telomerase, followed by caffeic acid and ferulic acid. So why this happens? According to their previous reports and the review, the number of hydroxyl groups or ring substituents in the molecule, it's a main factor in anti-cancer effect of these compounds. So it is still not clear why this happens, but it can be one of the reasons why they have different activity. Are stronger or different activity. So in this case, for caffeic acid, since it's a dehydroxylated phenol, and coumaric and ferulic acid are only one hydroxyl in their structure, they hypothesize that this can be an effect of or a factor that affects this inhibition of the expression. So increasing the concentration of the caffeic and ferulic acid, in this case, leads to elimination in the effects of these phenolic acids. So as you can see, if we increase for ferulic and caffeic acid, it leads to elimination. But if we increase for coumaric acid, this increases. So they still don't know exactly why this happens. It's only like an hypothesis for this. So coming back to the question, if these phenolic acids affect the human telomerase expression, yes, these results suggest that these molecules cause a downregulation of this gene expression. However, it's still not clear if this is dose dependent or if the structure is completely related to the intensity of the inhibition. So the mechanism is not clear. So they try to explain a little bit the mechanism or the mechanisms that can be involved in the human telomerase inhibition. As we can see, they reported that these phenolic acids cause reduced proliferation, and it seems that is caused by apoptosis. They also reported that these molecules reduce the human telomerase expression on HepG2 cells, but the inhibition level or intensity can be related to the structure of the phenolic acid, which can be also dose dependent. So they try to convey these results into a mechanism or mechanisms that can be related, but these are only questions or hypotheses of what can be happening here. So there can be different mechanisms involved in this expression and downregulation. So they first said that elevated concentrations of these phenolic compounds can play a role in pro-oxidant cells. So exposing cells to reactive species of oxygen leads to an increase in telomerase oxidation. So since these phenolic acids can have an effect, or if they are in low concentrations, they can have a relationship with reactive oxygen species. It means that the concentration of these phenolic acids is relevant for reducing the human telomerase expression. In the other hand, another mechanism that can be involved is the activation of the oncogene c-MYC, which is a modulatory factor in human telomerase expression. And they say that this gene binding region of the human telomerase might be the direct target of natural compounds, which in this case the phenolic acids might have anti-cancer properties through inhibition of the c-MYC transcription that is correlated to the human telomerase function. So even though these are only hypotheses, it is still not clear how this happens. So further studies are needed to elucidate the mechanism of action of these polyphenolic acids. So, like a brief summary, they saw that anti-cancer activity of phenolic acids can be mediated by reducing proliferation and also by downregulation of the human telomerase expression. And these three molecules, the caffeic, ferulic, and coumatic acids, can be promising natural products in cancer treatment. However, it's still necessary to explore the mechanism of action of these molecules. And as a personal comment, I think this article lacks different experiments to evaluate the effect of these compounds on the activity of telomerase. However, this is only the beginning of these molecules as a promising new compound for cancer treatment or even different therapeutic effects. So, yeah, in this case, I would like to end the presentation here. I will be very happy to receive your questions on November 3rd. And thank you so much.